Okay, students, so this is biosafety and bioethics, and the course code is BCHM315, and this is lecture 1, and the topic is the concept of biosafety. I am Dr. Musaddiq Ibrahim, Assistant Professor, Department of Chemistry, Pohart University of Science and Technology, and you can access these lectures through the Cust Media Server, whose address is www.ms.cohort.edu.pk. Now, moving towards the topics to be discussed in this lecture. So, first of all, we need to understand the definition of biosafety, then the importance of biosafety. Along with it, we will also discuss the biosafety issues, the biohazardous materials, and the pathogen risks assessment. Now, moving towards biosafety, that what is biosafety? So, biosafety is the prevention of large-scale loss of biological integrity and focusing both on ecology and human health. So basically, it is the prevention of biological material or biological agents not to be spread in the environment or to the persons, so that it shouldn't cause the harm. Now, the maintenance of safe conditions in the biological research to prevent harm to workers, non-laboratory organisms, or the environment. So basically, the researchers who are working in the lab, so they need to be safe from the hazards of the biological agents, as well as the non-laboratory organisms, so which means that other beneficial organisms which are present in the environment, so they should not be disturbed or damaged or killed by the biological agents with which the researchers are dealing in the laboratory. As well as the environment should be protected from the biological agents. So that is the concept of biosafety, and also the name itself explains that bio means life and safety means to get safe. So the life is safe from the infectious biological materials which can cause infection. Now, a fundamental objective of any biosafety program is the containment of potentially harmful biological agents. So when you are working in the lab, and for example you are dealing with E. coli species or Salmonella species of microorganisms, so the main objective, the main purpose of the laboratory is to keep the microorganism within the lab space and within the specified container or within the specified area, and it should not be going outside the lab, because if it goes outside the lab or the infection is spread outside the lab, so then it can cause infections to the other persons. Now, the term containment is used in describing safe methods, facilities, and equipment for managing infectious materials in the laboratory environment where they are being handled or maintained. So the containment is the following of or adopting of various procedures, methods, manuals, along with it the facilities which are available in the laboratory, so that when you are dealing with hazardous, biohazardous biological agents, so they are being contained inside the lab in a safe manner, so that it is not disturbing the laboratory environment, and they are being handled and maintained in a proper way. So there are some technical definitions related to the biosafety. First of all, they include the biohazard. So the biohazard is being defined as an agent of biological origin which has the capacity to produce or cause deleterious effects on human beings. So the examples are commonly the microorganisms, the toxins which are produced by the microorganisms, the allergens which are also the byproduct of the microorganisms, as well as the allergens and toxins which are derived or obtained from higher plants and animals. So all those agents which can damage or cause infection to the humans, and those biological agents are termed as biohazard. Now the biosafety, again you can see that it is the containment principles, the procedures, the standard practices, the technologies and the practices that are implemented to prevent the unintentional exposure of the pathogens and toxins or their accidental release. So as I discussed in the previous slide, so you can understand that what is the concept of biosafety is to keep safe the researchers, the experimenters, the lab workers who are working in the lab, as well as those people who are present outside the lab, by means of containing the biological agents in the specified containers or in the specified area. Now the term biosecurity, so it is also related to the biosafety, and what is happening in biosecurity is the control of accidental and deliberate release of biohazardous material. So if by some means the microorganisms, infection causing microorganisms have spread outside the lab or inside the lab, then there are certain protocols which are uh, encompassed by the biosecurity, and there are procedures by which you can control the spread of the infection. Either the infection has spread just accidentally, or somebody has deliberately released the biohazardous materials in the environment. So now we need to understand that why we need biosafety, and what is the purpose of biosafety, and what are the advantages of biosafety. So there's a background about the biosafety. So there were some unfortunate examples of disease outbreaks in the research labs. So in a period of time, some accidents they happen in the research labs, after which it was decided that the biosafety of the labs is very important. So for example, from 1950 to 1976, A survey of 5,000 labs showed that around 3,921 cases of disease outbreaks have taken place in the laboratories. Now, the most commonly reported cases were of hepatitis, tuberculosis, typhoid, brucellosis, and rabbit fever. So, these are very infectious uh, diseases, and they can even kill the human beings. From 2003 to 2014, uh, there was a SARS infect. The researchers in a lab in Singapore and a US and a Russian scientist, they were infected by Ebola virus. So one of them survived and one died. Similarly, a polio virus escaped from two Indian labs. That is also a very dangerous thing. And also a case appeared that scientists from Boston University, they contracted the rabbit fever, which is a very serious bacterial disease. Also, recently, the anthrax exposure in a Houston lab occurred due to the aerosols, the tiny droplets, which were leaked inside an unshielded centrifuge. So, by looking at the accidents which happened starting from 1950 up to 2014, you can imagine that those researchers or lab workers who are working in the lab, they are very vulnerable to the laboratory uh, practices. So in that case, the biosafety comes in, and there are strict practices and procedures which need to be followed inside the lab so that to avoid the spread of the infection. Now move towards that why we need biosafety and what are the advantages of biosafety. So first of all, by using the biosafety protocols, we can support the medical and scientific research on microbes and the human response to them. And furthermore, apply that specific type of research to the discovery and development of vaccines, drugs, diagnostic tests, which are designed to protect the general population. So when the researchers who are working in the lab, they are safe and sound, and they are dealing or handling infectious agents, and after doing their research work they come up with results. So these are the fruits of their results. But if the lab workers, they are not safe, then all these things are not possible. So the biosafety, it ensures that everyone has sufficient research facilities to carry out these activities. So in case of biosafety protocols, procedures, and practices, for them you need certain procedures, certain equipment, certain area, infrastructure of the lab. So if that is available, then the researcher, he or she can carry out the experiments. So now move towards biosafety in various disciplines. So the biosafety is related to several fields in science. So they include ecology, agriculture, medicine, chemistry, and biochemistry. So you can understand that all these fields, they need the laboratories, and when all these laboratories they have the lab workers working in them, so then the biosafety is important for all those lab workers. Now, biosafety in academic research. So we know that there is extensive research going on in universities, and as a result of the application of biosafety in the laboratories, it promotes the safe laboratory practices. As in the image, you can see that a researcher is working uh, within the biosafety lab, and he is working inside, uh, dealing with the microorganisms within the biosafety cabinet, and then you have a biohazard sign available onto the biosafety cabinet. Now, as a result or benefit of the biosafety, the procedures, the proper use of containment of equipment and facilities become possible. The biosafety, it also provides advice on laboratory design, that how the laboratory structure should be. Along with it, the risk assessment of experiments, that what are the risks which are posed by the experiment, and they should be taken care of, and those experiments which involve infectious agents, recombinant DNA, either in vitro and or in vivo. So the bottom line is risk and containment. So as a result of biosafety in the lab, first of all, the risks can be minimized and they can be known, as well as the containment, that if an infectious agent has been spread either inside the lab or outside the lab, so then how to contain, how to uh, control the spread of the infection is through the applications of biosafety. Now moving towards the biohazard symbol. So the biohazard symbol was introduced by Charles Baldwin at National Cancer Institute at National Institute of Health in 1966. So here, this is the biohazard symbol, and the symbol can be memorized. It has meanings, so it could be learned. And here in this image, you can see that the color of the biohazard symbol is blaze orange, which is most visible under harsh conditions, which means that if a biohazardous material is present in a container and that container is being labeled with the biohazard symbol, and that container is present in a snowy area or a rainy area, but the color is so visible that it can be seen by the general public or the lab workers. Now, based on the biohazard symbol, there have been basic classes of laboratories risk derived from low to high. So first of all, you have BSL1 laboratories, then BSL2, then 3, and then 4. And according to the level of severity of the uh, danger of the biological materials or agents, you can see that the color is also changing starting from BSL-1 up to BSL-4. Now, if you look at the symbol of the biohazard, so you can see that there are four circles. So one circle is in the center, and then there are three circles around it. So a total of four circles. Now these four circles, they represent the one circle represent the agent. So the agent can be the pathogen, the bacteria, the virus, which can cause the infection. The second circle, it stands for the host. So the host can be a lab worker, a general public member, uh, someone who is working in the lab, a researcher. The third circle stands for the source. So it means that what is the source of the biological material, whether it came from a container or it came from the air through breathing, or it came from the transmission through contact with the surface or with the person. As here nowadays we can see that the coronavirus is spreading through uh, person to person by shake hand or by hugging each other. So it means that these are the sources. And the fourth circle stands for the transmission, that whether the transmission, it can take place through air or by contacting the surface where the pathogen is present. So these are the uh, four circles and their representation as agent, host, source, and transmission. So now moving towards the biosafety issues. So the issues include that the laboratory safety. So as a result of applying the biosafety rules, the laboratory safety can be guaranteed. Now there are blood-borne pathogens with which you are dealing within the biosafety labs. Also you are dealing with recombinant DNA, that is rDNA. For example, in case of testing for hepatitis A, B or C virus, you have the recombinant DNA available as a sample for proliferation of the hepatitis A, B or C virus DNA. Also, biosafety is related to the biological waste disposal, that how the biological waste disposed into the environment in a secure manner, uh, as well as the infectious substances and the diagnostic specimen shipping. So these are the issues which are related with the biosafety, that safety precautions should be adopted when dealing with these issues, so that the danger or accident should not happen as a whole. Now, continuing the biosafety issues, so some of the issues include the respiratory protection. So whenever you are working in a BSL 1, 2, 3 or 4 lab, you should be wearing a proper mask, so that the infectious agents should not be inhaled. There is a safety issue of bioterrorism and select agents. So there have been examples that in some cases the infectious agents have been deliberately spread out into the environment so that the people can get infected especially in case of wars. The outdoor and indoor air quality is also a very important biosafety issue, so that should be maintained as well. And occupational safety and health in the use of research animals. So in many BSL labs, the research animals like mice are being used. So the safety precautions, the procedures, the standard practices should be adopted so that to avoid any issues arising from them. So in this picture you can see that a researcher is injecting some medicine into the mice, and the researcher is wearing gloves, um, also wearing a protective equipment as a whole, which is covering the hairs, the lower body. So these are the precautions which need to be taken so that to avoid the biosafety issues. And also biosafety issues are present in case of biohazards used in animal models. So if animal models or animals are being used in the lab, so there is a possibility that that animal is being infected with certain disease. So in that case, there is a biosafety issue which need to be taken care of. So biohazardous materials or pathogens. So these include the viruses, bacteria, fungi, chlamydia or rickettsia, prions, and recombinant DNA. So these are the pathogens which are under observation or which are being analyzed by the researchers, by the lab workers when working in the lab, and they are termed as biohazardous materials because these are disease causing agents. Now the biohazardous materials other than virus, bacteria, fungi, and others which we have discussed in the previous slide, there are certain others materials which are also termed as a biohazardous material. So for example, the transgenic plants, animals, and insects. So transgenic means that those plants, animals, and insects which have been genetically modified. Here you can see that the case of mice who have been genetically modified, so it means that they are termed or included as biohazardous material. Now other biohazardous material, they also include the cells, tissues, and body fluids of humans and primates. So if a human being or a person is infected or is suffering from a disease, so his or her cells, tissues, and body fluids, they will be termed as biohazardous material, as well as the primates, for example the monkeys, their cells, tissues, and body fluids will also be termed as biohazardous material. Now other biohazardous material include the brain tissues from the demented patients. So demented patients means that those patients who are mentally ill or who have a degenerated brain, as well as the viral vectors. They are also included in the biohazardous materials because the viral vectors they can carry as well as they can transmit a specific disease or infection.